Thank you. Good afternoon, everyone. I would like to thank Dr. Keshav Singh and Dr. Thakarajan for inviting me. Such a great place, such a great conference. Thank you so much. I'm enjoying with all my colleagues from US. So today I'm going to give a talk on microRNAs and prostate cancer and tell you a little bit about what my lab has been doing since last few years. Basically, I'll talk about these three main areas and give you some snippets, some highlights, short stories about these three areas, why I study prostate cancer and prostate cancer disparities, and how I started. What non-coding RNAs, microRNAs are, because that's what I work on, how they modulate gene expression in prostate cancer. And I'll also talk a little bit about microRNA profiling, which is basically a tool that we use to study microRNAs in human samples. So that's what I'm going to be doing. So in terms of prostate cancer, the incidence and mortality of prostate cancer incidence is pretty high in the West, but if you think about incidence, Australia and New Zealand actually ranks highest in terms of prostate cancer. And then comes the US, but mortality rate is not as high in US, maybe access to healthcare or whatever it is, but it is very high in some of the African countries, as you can see over here. Why prostate cancer is important? Although incidence and mortality is declining over here, but the gap remains between the African Americans and the other populations, and that's the reason why I think it's very important, and prostate cancer is a disease of disparity. With African American men having a higher incidence of prostate cancer. So I started my journey from Lucknow and over here, and tuberculosis is highly prevalent in India, and again India was the catchment area when I did my PhD. So I did my PhD with on tuberculosis at CDLI Lucknow, Chatar Manzil, and then I moved to Washington, D.C., and started my journey in Georgetown in North America, which has very high incidence of prostate cancer, if you can see over here. And then I moved to UDC, which is University of D.C., another university in D.C., and where I continued to work on prostate cancer. So I'll give you a little bit about how prostate cancer looks like in Washington, D.C., where I was. Again, Washington, D.C., when you join a place, I mean, you basically serve the people of where you are. So prostate cancer disparities are prevalent, and the incidence and mortality rate is high in Washington, D.C. Washington, D.C. Has the highest prostate cancer mortality rate in U.S. And for my friends who are from US and from DC area, they will appreciate that, and they know that already. But there is a very sharp divide in Washington, DC. This is the DC map, and this is the road that we call 16th Street. So if you look at 16th Street, you can really see the difference of people who are living on this side and who are living on this side. So if you look at this, the incidence rate is 128 over here, and incidence rate 236 over here. So there's a very sharp contrast with prostate cancer incidence in DC. What else is going on in DC? Ward 6 has the highest incidence rate of prostate cancer in DC over here, and the mortality rate is highest in Ward 1. So DC is divided into 8 different wards. And if you look at who lives in those wards, look at this over here. So it is predominantly African Americans who live on the other side of the river, as we call it, from 16th Street. Low education, low income, and look at the high incidence of crime rate in DC, in these areas. And if you overlap it over here, basically, so if you see this Ward 1 over here, which has the highest mortality of prostate cancer, is it due to that? No, it is not. But it does affect the living conditions of people who live there. So, I mean, it is much more complicated than just the race. That's what I'm trying to get to. So moving on, what I work on is RNA molecule, and which is microRNA. And not too long ago, when we used to study three kinds of RNAs, and now look at what the diversity of RNA molecules. And today we are going to talk a little bit about microRNAs over here. What microRNAs are? MicroRNAs are small non-coding RNA molecules. They repress the activity of messenger RNA by binding to the 3' UTR region. They estimate about 30% of mammalian gene products. Depending on who you ask, it is much more. One microRNA can target hundreds of messenger RNAs, which is a good thing and a bad thing, again, depending on who you ask. The bad thing is that the reviewers are going to tell you that, oh, it's not specific. The good thing is that they may say that, look, one microRNA may regulate the whole pathways by modulating different genes in the same pathway. So why study them? MicroRNA, I think, is the manageable snapshot of functional human genome. If you do genes versus genomes or whatever you do, you have to do 30,000 genes or 3 billion base pairs. But if you do the whole transcriptome of microRNAs, you can get away with 2,000 microRNAs approximately. Highly stable. Our hospitals are banked with a lot of FFPE samples and body fluids, so you can detect microRNAs very easily. So that's a good advantage of studying microRNAs. They are fine tuners of gene expression, and they can be modulated by environmental factors. A bit of microRNA biogenesis. Just like messenger RNA, they are transcribed in a similar fashion by RNA polymerase 2 and 3 as a primary transcript. They are cleaved. They have a hairpin loop there, and then they become precursor microRNAs because they are cleaved over here by DGCRA or DROSHA, and then they are transported out. This hairpin loop is cleaved by dicer, and then one of the strand of microRNA, this is degraded depending on the condition, again, which strand is going to be degraded, and the mature microRNA goes and binds to messenger RNA and does whatever it is supposed to do to repress gene expression. Just like genes, depending on whether microRNAs are up or down, they could be oncomiRs or tumor suppressor miRs, and they regulate various cancer hallmarks, which I will show you in a little bit. Profiling, I mean, this is an important thing because think about it. I mean, if you want to do population-based studies, you are basically relying on human samples. So it is important to acknowledge the importance of microRNAs because you can detect microRNAs very easily in human samples and FFPE tissues, cell lines. People have done, but plasma, serum, and PAX gene tubes that we collect. So these are various methods that people have been using, and we have used basically the HTG technique, the microarrays, the microarray, which was the old technique, the NGS, and the qPCR. Cost-wise, these are different methods that people have been using. We started with microarrays and RT-PCR. Now we did then NGS, next-gen sequencing, to do the microRNA expression analysis. Right now we are at the HDG-seq, which is a pretty good platform, and we recently established a core facility to do the microRNA analysis by HDG-seq. And what it does is it basically doesn't do microRNA SNPs or variations, but expression levels. It's a very powerful technology that we are trying to utilize. So just like other studies, we started our studies not too long ago. We took the prostate cancer tissues, and we started with the laser capture microdissection, looking at the benign and the tumor tissues, and we did the profiling using the microarrays and the RT-PCR arrays, which was the PCR array by Applied Biosystems, and we found various microRNAs which were differentially modulated between the benign tissue and the prostate cancer tissues. Here's an example of 4 of the microRNAs that were modulated in these tissues. Looking at the race, one of the microRNAs, and this is a microRNA that I'm going to give you an example of. Once you identify these microRNAs, what can you do? What's next? So basically, just like any gene studies or functional studies, you take that microRNA, you characterize them, and you hope that whether you can use it as a biomarker or as a molecular target, and also to study the signaling mechanism. So microRNA 214 is differentially modulated in African American prostate cancer, as you can see here. MicroRNAs, they modulate various genes. So basically, if this is really downregulated in African American, that means that the genes it targets must be upregulated in African American. So we look at the GSC data, and we found that there were multiple genes which microRNA 214 targets, and all of those were upregulated in African Americans, that you can see the dark bars over here. So that gave us some confidence that this microRNA may have a role in cancer and may play a role in the disparities. Patrice Cagle, who is a postdoc in my lab, she tried to characterize, and this is just again a snapshot of what you do when you do identify the microRNA. Not going into detail, but you take the microRNAs, you overexpress knockdown, and you study whether it modulates different cancer hallmarks or not. Again, angiogenesis, NOIK cell cycle and proliferation and things like that. So we did that with this microRNA, again the colony formation and all those things. So this is just to show that the microRNA, which was differentially modulated, does have a functional role in modulating cancer hallmarks and could be exploited as a therapeutic target or whatever you want to do. Here is another microRNA, which I'm going to show over here, the microRNA 99B, which was also modulated in a very similar way in African Americans more than Caucasians. But this particular microRNA targets a transcription factor, SP1, and all these and SP1 targets, some of them were upregulated in African American prostate cancer. Again, just a little confidence in terms of that, yes, it may have a role in there. We did characterize all these microRNAs further, just like the 214 that I described, so I'm not going to go in detail of those characterizations, but the important thing that I do want to share with you is that both microRNA 214 and 99B, they target a protein tyrosine kinase, which is PTK6, and which was pretty interesting. So we found that this, and PTK6, so this is how you see, you take the microRNA, you do the algorithms, you do the in silico studies to identify what could be the potential target of that microRNA, and then you do these studies to see whether the protein is modulated or not, and then you do the luciferase assay to show that yes, indeed, this microRNA may bind into 3' UTR region of that gene. So we did that. PTK6 is known to be involved in prostate cancer. High levels of PTK6, they do predict poor prognosis in patients with prostate cancer, and like for example over here, metastatic prostate cancer has high levels of PTK6. If you have metastasis, then metastasizing tumors, they have high levels of PTK6, and as you reduce the expression of E-cadherin, then you have a high levels of PTK6. So we were excited about this PTK6, and in a related but a different study in my lab, we were trying to also look at known cancer drugs to identify new targets. And what we found was ibrutinib, which is known to be used in liquid cancers, and it targets a kinase, which is Protein Tyrosine Kinase. We were doing the study, and what we found was that it does also targets PTK6, which is the BRK over here. So, I mean, that was an interesting thing. We published it last year, but the idea is that if these microRNAs are downregulated in prostate cancer, that means they can target that means that PTK6 would be upregulated over here. So can we stratify prostate cancer patients based on microRNA expression and or PTK6 expression, and maybe those could be targeted by ibrutinib. So that could be one of the example of how you can utilize these microRNA expression studies. Or, just like everything else, you can use it for microRNA therapeutics. So microRNA therapeutics, just like gene and expression studies, if the microRNA is overexpressed, then you identify the antimeres. And if it is downregulated, tumor suppressor microRNA, then you go with the microRNA replacement therapy. So you can do that, and people have used various methods to really deliver those microRNAs, because one of the problems, similar to many other oligos, is that delivery is a big deal. So people have identified ways to deliver those microRNAs, ranging from liposomes to exosomes to nanoparticles. So I mean, that could be another forward-looking thing in terms of what you can do once you identify a microRNA of your choice. Switching gears, we're talking about disparity, and one of the things that my lab has been interested in is like, okay, so what else is involved other than race when you talk about the disparities? And one of the important piece is the stress and the living conditions. And there is a good theory which is social signal transduction theory of depression. And I mean, you can use this theory in terms of just like anything. So you have social stresses, they do affect all these pathways in the body. For example, the HPA pathway, the SNS, or the vagus nerve, they all release all these hormones that can go inside the cell through these receptor molecules and then alter the gene expression or the microRNA expression and all those things. So people have been studying access to healthcare, social determinants, but the linkage from social determinants with this has been missing a little bit. And as I showed you before, I mean, there is a good amount of data in terms of economic stability, education, neighborhood environment, health and healthcare, and social and community context, which you can translate to these hormones. I mean, think about it. It has been shown that people who feel isolated, they have lower levels of IL-6. So, I mean, and as Anil was showing yesterday and other people have shown that, I mean, IL-6 does have biological consequences. So does it mean that people people who have these differential social status or differential perceived social is happening over here? So average annual spending on fruits and vegetables. So these people, they don't spend money. These are the these are the areas where you have predominantly African Americans, higher incidence of prostate cancer, the crime rate, below levels of poverty. I put this slide over here, although these are the new houses over here, these are the old houses, but these are the affluent old houses that people take and renovate them. So there are a lot of things that you can do. So what my lab has been doing is we are collecting serum samples from different regions of Washington, D.C. So if you collect all these samples, then you can really study that can we identify microRNAs related to neighborhood factors. So I think that's what, and the advantage of doing all these things is that you can maybe study whether all these social factors may modulate cancer hallmarks through these microRNAs, maybe. Or, I'm not going to go through this slide, but we all know the prostate cancer, or can we identify and use microRNAs as prognostic markers? So where I'm going is that we talked about the tissue microRNAs, but in this case, we also did some circulatory microRNAs which were present in the blood. And we identified several microRNAs which were differentially modulated in prostate cancer when you compare with the normal individuals. And this particular expression is differentially present between Caucasians and African Americans. Sometimes the difference is more profound. Sometimes it is not more profound. So again, the difference, you can study these things to really make sense in terms of what is basically happening. So when we talk about social stress, as we said, that what happens to people? What happens to people is they have hypervigilance, anticipation of social anxiety, sleep, depressed mood, you eat more, obesity, accelerated aging, and prostate cancer is a disease of aging. So these are some of the thought-provoking things that my lab is trying to figure out. I mean, think about it. Caucasians and African Americans, look at over here, the circadian period is different between the races. African Americans, Caucasians, and Asians. So over here. And this may not look more, but this has some profound consequences over here. So what we did was we looked at some of the hormones which were present during sleep, during obesity, and cortisol, and we found that many of the microRNAs that we were studying, they were indeed differentially modulated by these hormones that we are trying to study. So, and what we are also trying to do is that when we have these microRNAs which are circulatory, we are also trying to find out what does it mean, what is the mechanism. Are they involved in cell-cell communication? Are they just biomarkers? Where are they coming from? Are they really going to modulate the immune landscape, as Anil was saying about the JAGED1 pathway earlier? So one of the molecule that we have, microRNA 628-5P, for example, it targets JAGED1. I don't have the slide to show you, but it is a circulatory molecule. If it is a circulatory molecule, then maybe if it is present in the blood vessel, then maybe it could be modulating JAGED1 on those dendritic cells that Anil was showing, or it could be modulating JAGED1 in the endothelial lining of these circulatory microRNAs. And what is happening is that these JAGED1 could be disrupting this paracrine signaling between the immune cells or endothelial cells and the tumor cells, thus modulating these cancer hallmarks. So, I mean, so once you have that, then you have these molecules. Those could be modulated by these environmental factors, hormones, or disease per se, but they may have profound effect inside that. This is just an example of very recent data that we have received, that a lot of times you can get data from individuals or samples, that where are they from, whether you have social data or not. A lot of times you don't. So can you use these hormones as surrogates of those? So what we did was we identified these samples and we did the leptin high and leptin low. And we found that between leptin high and leptin low, there are a lot of microRNAs which were differentially modulated in this. So you can do these kind of studies to really make sense in terms of what is happening with this. So summarizing, they are differentially expressed in prostate cancer and African American prostate cancer. Tumor suppressant microRNAs may modulate cancer hallmarks and a potential candidate for microRNA replacement or microRNA therapy. Social stress-related hormones may function through microRNAs to modulate cancer hallmarks, and they could be modulated as the targets. Here's my institute with 22 teams who are working on health disparities issues. Here's my team, and I do thank a lot of folks who have helped me with this study. So, and I'm happy to take questions.
Yeah.